To begin with, the man given credit for viewing this invisible world for the first time was not a trained scientist. Anton van Leeuwenhoek was a Dutchman who ran a dry goods store selling cloth. Was the first person to develop a microscope powerful enough to view very tiny organisms. This is what one of his common single-lens microscopes looked like. Yet some could magnify things as much as 300 times. He used this simple microscope to look at all kinds of things. He peered at a bee's stinger, moldy bread, hair, blood, cloth fibers, the head of a fly, dust, and parts of plants. He kept careful notes and drawings of what he saw. However, his greatest discovery came when he decided to look at a drop of pond water. He saw little creatures swimming around in the water. He was the first man to see living creatures too small for our eyes to see. We now call these creatures microbes, and therefore Leeuwenhoek is known as the father of microbiology.
As powerful as the stereo microscope is, much more powerful is a standard light microscope. Most student light microscopes usually have a single eyepiece lens. The objective lens is usually located on a rotating turret with several other lenses. Multiple objectives provide a variety of magnifications. This objective will magnify an object 4 times. Its power is 4. This objective will magnify an object 10 times. Its power is 10. Finally, this objective will magnify an object 40 times. Its power is 40. The eyepiece of a typical student microscope usually has a magnification of 10. When you combine a 10-power eyepiece with a 4-power objective, the overall power or magnification becomes 40. This is a brine shrimp magnified 40 times. At this power, you can get an overall view of an object. When you combine a 10-power eyepiece with a 10-power objective, the overall magnification becomes 100. This is the brine shrimp magnified 100 times. Now we can see details we weren't able to see before. These are its eggs. When you combine a 10-power eyepiece with a 40-power objective, the overall magnification becomes 400. These are the brine shrimp's eggs magnified 400 times. Each magnification teaches us something new and different about a specimen.
The electron microscope consists of a cylindrical tube about 2 meters long, which is completely devoid of air. In contrast to the light microscope, the electron microscope visualizes objects using a thin beam of rapidly moving electrons that interfere with the specimen placed in the tube. The electrons are emitted by the cathode at the top of the tube and then accelerated by the anode. They then pass through a small aperture, which forms them into a beam, and into the vacuum inside the tube. The part of the microscope that generates the electron beam is sometimes called the electron gun. The beam is maintained along the tube by means of electromagnetic lenses. These are coils that surround the tube at given intervals. The electromagnetic field emitted by the coils focuses the beam at the center of the tube. Electrons encounter the specimen and are either absorbed, scattered, or pass through it. Because different regions of the specimen are variously transparent to electrons, different amounts of electrons with changed energy pass through these regions. At the end of the tube, the electrons are collected on fluorescent or photographic film, or on screens that generate an image of the specimen. The beam that reaches the film consists of the different amounts of electrons that pass through particular regions of the specimen. This difference is responsible for the contrast in the film. The original image produced by the electron microscope is always black and white, and it is not possible to see it directly with the eye.